pseudoheptulose. By bisphosphate, it loses a phosphate group and forming the pseudoheptulose 7 phosphate. So the enzyme is the phosphatase. Pseudoheptulose is the 7 carbonous substances, Nana. Just in this way, if you want the almost all the reactions, this is the complete the fourth reaction. Erythrose 4-phosphate plus dihydroxyacetone phosphate, pseudoheptulose 1,7 diphosphate. So erythrose is 4 carbon, dihydroxy is the 3 carbon, 4 plus 3 7 carbonous in the presence of aldolase enzyme. So 2 phosphates are adding as it is in the pseudoheptulose 1,7 diphosphate. So
Madam,
this
if
lose
possible,
1 phosphate
enlarge.
group in the presence of sir. Okay, sir.
Okay. Okay. Right.
So diphosphate, it will lose 1 phosphate group in the presence of the enzyme phosphatase to form the pseudoheptulose 7 phosphate. This is the complete 4 plus 3 convert to the in the presence of aldolase enzyme. Both will combine to form pseudoheptulose 1,7 diphosphate, di or biphosphate as your choice. So the pseudoheptulose 7 phosphate, it react with 3 phosphoglyceraldehyde in the presence of trans-ketolase enzyme to form the xylose 5-phosphate and ribulose 5-phosphate. Just to this is the just you can observe here the pseudoheptulose 7 in the presence of trans-ketolase enzyme and convert as a xylose 5-phosphate plus ribulose 5-phosphate. So pseudoheptulose is 7 carbonous, phosphoglyceraldehyde is the 3 carbonous. So 7 carbonous compound will split into xylose 5-phosphate 5 carbonous, ribulose 5-phosphate is the 5 carbonous compound. Okay. So this is the almost to the end product of the ribulose 5-phosphate and it to combine with utilize the ATP energy. So in the presence of phosphopentokinase enzyme and formed as a ribulose 1,5 diphosphate. So in this almost the xylose 5-phosphate is converted isomerized to ribulose 5-phosphate in the presence of enzyme and another way epimerase also. So the ribose 5-phosphate is converted into ribulose 5-phosphate through the action of the pentose phosphate isomerase enzyme, phosphopentokinase or pentose phosphate isomerase enzyme. So in this, this is the final reaction of the RUBP regeneration. Finally, the ribulose 5-phosphate is converted to ribulose 1,5 biphosphate through the action of the ribulose phosphate kinase, phosphopentokinase. Okay, and ATP also utilizes in this final reaction. So this is the two molecules or 2 moles. 5 plus 5, so almost 2 moles in the end product. 5 plus 5 ribulose and ribulose 1,5 diphosphate also formed RUBP, ribulose 1,5 biphosphate. So RUBP also regenerated in the last reaction of the RUBP regeneration step. Okay, so for each molecule of carbon dioxide fixation, CO2 fixation and reduction, 2 NADPHs and 3 ATPs are required. So we know in light phase ends, this is the cycle. This is the assimilation or the total cycle of the Kelvin-Benson cycle. So in the last, the significance of C3 cycle or the assimilation power of the C3 cycle for each molecule of CO2 fixation, almost 2 NADPHs and 3 ATPs are required. So it can be concluded that the 6 tons of the cycle, that means one glucose is formed 6 C6H12O6. So it can be concluded that 6 tons of this cycle are required to produce a hexose phosphate. So this reaction can be almost in this, just I will show that only the split of the cycle 6 CO2 nana, almost all you can double that 6 CO2, 10 H2Os, 12 NADPHs, 18 ATPs are utilized in this reaction and it converted as a glyceraldehyde 3-phosphate. Otherwise, so if we will take 6 carbons, so that is the glucose C6H12O6. Instead of glyceraldehyde 3-phosphate, we'll put into C6H12O6. Then the double the ante 6 CO2s, 10 H2Os, 12 NADPHs, 18 ATPs. So these all are utilized in this net reaction. It can be converted as a one molecule of the C6H12O6 plus 12 NADP plus 6 H pluses plus 18 ADPs plus 18 inorganic phosphates. So that is the net reaction of this cycle. So almost all the fine, but what we will take this, all the steps in this, just to I'll share this cycle. You can observe here all these. So only will take that 3 CO2s, the final reaction. Now I take 3 carbonous compound. So that is the almost in the okay. So that is the 6 carbons are entered. The phase 1 carbon fixation, Rubisco, and 3-phosphoglyceraldehyde. So in the 6 ATPs are utilized. 18 ATPs where it utilized in this reaction. So first step, almost in the reduction phase, how many ATPs are utilized in the reduction phase, Nana? So okay, so in the reduction phase, almost 6 ATPs are utilized in this phase, and 12 ATPs and 12 NADPHs. 12 number will utilized in the reduction phase, and finally the almost remaining 6 ATPs are utilized in the last RUBP regeneration phase. Okay, so that is finally the RUBP is converted to ribulose 1,5-phosphate through the action of the ribulose phosphate kinase, and ATPs also utilized to convert as ATP. 18 ATPs plus 12 NADPHs are the utilized in this way. So generally for formation of one glucose molecule, one glucose means C6H12O6. So for formation of the one glucose molecule, 6 CO2 molecules are required. Okay, so for formation of the net reaction, just to I'll explain that the formation of one glucose molecule, 6 CO2 molecules are the required. For one CO2 molecule to undergo the reduction, 3 ATPs, 2 NADPHs are required. So 3 ATPs, 2 NADPHs are we are benefited from during the light reaction, during photophosphorylation, cyclic and non-cyclic. The assimilation power of the cyclic non-cyclic will get into 3 ATPs, 2 NADPHs. So for that purpose, for one CO2 molecule to undergo the reduction, 3 ATPs, 2 NADPHs are required. One CO2 molecule when participates 1 in Kelvin cycle, 1/6 glucose is formed. Okay, catch it, Nana? So for formation of one glucose molecule, 6 CO2 molecules, 6 carbon dioxide molecules are required, and 18 ATPs, 12 NADPHs are required for the one glucose molecule formation. So this is the complete net reaction of the C3 cycle. Okay, so here that means the hexose formation and sucrose formation occur. These all are in the cytosol. Since phosphoglyceraldehyde are transported out of the chloroplast via antiport carrier system. So in this, the phosphoglyceraldehyde and dihydroxyacetone phosphate both will condense under the influence of enzyme aldolase. They will forming the fructose 1,6-bisphosphate. So the removal of phosphate by enzyme phosphatase results into fructose 6-phosphate. So the fructose 6-phosphate is converted to glucose 6-phosphate. In this, almost for our convenience, almost the categorized phosphoglyceraldehyde, dihydroxyacetone phosphates, these all are for our convenience. We will split into minimum 5 groups. So in that, the 12 phosphoglyceraldehyde, just I'll check it. In the phase, numbering are not mentioned here. The phosphoglyceraldehyde and dihydroxyacetone phosphate. So almost in the reduction phase, for 6 molecule of carbon dioxide will convert into 1 glucose sugar. So that is almost to 12 phosphoglyceraldehyde. 6 times cycle jaragali. Andukani manaki ikkada numbering anedi just nenu mention cheyaledu. మీకు ఇక్కడ ఏదైనా 3 phosphoglyceraldehyde can convert into dihydroxyacetone phosphate in the presence of isomerase. So almost the 12 phosphoglyceraldehyde can splitted into 2 phosphoglyceraldehyde, 2 dihydroxyacetone phosphate, 2 phosphoglyceraldehyde, 3 dihydroxyacetone phosphate, 3 phosphoglyceraldehyde. For our convenience only, we will take this. So in that, the intermediate substances like erythrose 4-phosphate, xylose 5-phosphate, pseudoheptulose, ribulose phosphate, xylose phosphate, these all are the intermediate substances. They are the unstable compound. And finally, the last phase of the ribulose biphosphate is utilized the ATPs. So 6 ribulose phosphate is utilized 6 ATPs. They can convert into, or they can regeneration the ribulose biphosphate RUBP regeneration. So almost the cycle is continuous. This 6 RUBP combined with carbon dioxide, atmospheric carbon dioxide, undergo the fixation first step. After that, phosphoglyceraldehyde is formed. So this is reduction phase. After that, RUBP, it is undergo the continuous cycle to form the sugars. Okay, so that's why the photosynthesis, not only the it is involved in the almost interdependent to light reaction to the dark reactions. So in the dark reactions, this is any regard nana almost studied in your syllabus, but any ATPs are any doubt, please ask me. What is the importance of C3 cycle? So almost we'll discuss. That is the completely, it is the Mamata. So the plants, they are almost they in which CO2 is initially fixed as the 3-phosphoglyceric acid. So the Kelvin cycle occurs in the stroma of the chloroplast. So it is, this shows that the Rubisco enzyme is mainly key enzyme in this way. So the cyclic pathway that uses the phosphoglyceric acid. So it is a C3 compound nana to form the other sugar phosphates and regeneration of the RUBP. So all will collectively known as that Kelvin cycle. So it is the Kelvin and Melvin cycle also. In some ways, it is also Kelvin-Benson cycle also. So it consists of the carboxylation phase, reduction phase, and regeneration phase. So these all intermediate substances will the biochemical or biocatalysis reactions are occur in the RUBP regenerations. The number of series will occur in this. Finally, the 6 carbon dioxide, the assimilation power that for each molecule of CO2 fixation and reduction, 2 NADPHs, 3 ATPs are required. So it can be concluded that the 6 tons of the cycle are required to produce a hexose phosphate. That means glucose or fructose or any C6H12O6. So that is of the 12 phosphoglyceraldehyde molecules, they all are generated by the reduction of 6 molecules of the carbon dioxide. Okay, so in this almost the remaining 2 molecules, for our convenience, we will split it into 5 different ways. Almost other, I'll share other diagram. Just you can just to one neat diagram will, I have that, just wait, Nana. Tirupati sir, are you there? Oh. Just wait. Hello, sir. Am I audible to you? Hello. Hello.
Yes, ma'am.